Hello and welcome to this BioProcess International Ask the Expert webcast. I'm your host, Leah Rosen, the online editor for BioProcess International. Before we get started, just a couple of notes. This webcast is being recorded and will be made available for replay in the multimedia section of our website. We've muted the audio lines, but we welcome you to type in your questions for our speakers in the question and answer window on your screen. After the presentation, we will begin the question and answer portion, and I will ask our speakers your questions from the question and answer window. Today's webcast is actually two separate presentations. The first is by Dr. Roland Herzog, Professor of Immunology and Director of Gene and Cell Therapy Program at the Indiana School of Medicine. The second will follow directly and will be given by Dr. Nagendra Venkata Chemuturi from Takeda.
Adeno-associated viruses, or AAV, are some of the most widely used vectors for transfer of therapeutic genes in treatment of human disease by gene therapy. So I'd like to thank the organizers for giving me the opportunity here to present to you some of our thoughts and some of our work on how AAV gene transfer interacts with the immune system. We're keeping in mind that immune responses are some of the greatest hurdles for successful gene therapy. AAV vectors are primarily used for in vivo gene transfer, with the viral capsid determining the tissue tropism, and given the wide range of choices of AAV capsids, we can appropriately design the vector to achieve high levels of gene transfer to a particular target cell or a target tissue. Within this viral capsid that is made up of viral proteins, a single-stranded DNA genome is contained that is less than 5 kb and can also be engineered to be a self-complementary DNA genome. Currently, AAV-based gene therapy drugs are approved for the treatment of two genetic diseases. One is a form of blindness, and the other one a neurodegenerative disorder called spinal muscular atrophy. Furthermore, phase 3 clinical trials are being currently conducted for the treatment of the inherited bleeding disorders, hemophilia A and B, and for another muscular disorder called X-linked myotubular myopathy. Despite these successes, hurdles of therapeutic applications of AAV gene transfer have also emerged. These include the potential for immune responses against either the vector or its therapeutic gene products. This slide summarizes some of the immune responses that have been observed in clinical trials. These include lack of gene transfer in patients that have pre-existing neutralizing antibodies as a result of natural infection with AAV viruses, CD8 T-cell responses against the viral capsid have been observed in liver and muscle gene transfer. There has been examples of hepatotoxicity, which was observed in liver-directed gene transfer as well as systemic gene transfer, where the virus is given intravenously for transfer to multiple organs. In some of these cases, hepatotoxicity can be attributed to immune responses. In other cases, it is less clear what the cause of the toxicity is. At very high systemic vector doses, complement activation has been observed, and in some cases, additionally, activation of platelets. CD8 T-cell and antibody responses to transgene products have been described in some cases in muscle gene transfer, and ocular inflammation has also been described. And therefore, the addition of various types of immune suppression and modulation regimens has been a theme in the development of clinical AAV gene therapies. And also there are changes to vector designs that are specifically prompted by observations of immunogenicity in an attempt to make the vectors less immunogenic. I'll show you some examples of how you might be able to accomplish that. Here are some of the molecular structures that the immune system can recognize during AAV gene transfer. For example, the immune system has innate pattern recognition receptors that can potentially sense the viral capsid or the viral genome. We know that upon gene transfer, the immune system within days is developing neutralizing antibodies that bind to the capsid and prevent readministration. And we also learned that the viral capsid can be subject to CD8 cytolytic T cell responses. Furthermore, it's become more clear recently that at very high systemic vector doses, AAV administration can result in activation of complement. The data presented at various recent meetings suggests that it is primarily the classical pathway that results in complement activation, which requires antibodies to bind to the AAV capsid and cause antibody-mediated complement activation. However, one of the complement components, C3b, is also known to bind directly to the capsid, which may not necessarily result in activation of complement, but would certainly drive increased opsonization of AAV particles, which also may enhance immunity. Upon gene transfer and transgene expression, of course, adaptive immune responses in form of antibody and T cell responses specific to the transgene product may occur. And it's recently been proposed that the virus also expresses some level of double-stranded RNA. The mechanism resulting in this double-stranded RNA formation is not entirely clear yet. But if that really were to happen, then this could be another source of innate immunity through sensing of double-stranded RNA by pattern recognition receptors. Now, we know that sensing of the AAV genome through toll-like receptor 9 results in tissue inflammation and type 1 interferon production. And so the question that we wanted to answer is whether the innate immune sensing of AAV translates into adaptive immune responses, or in other words, how do innate immune responses link to antigen-specific adaptive immune responses? I want to start addressing this question through some observations that came out of gene therapy trials for the X-linked coagulation disorder, hemophilia. Now, patients are treated through intravenous infusion of coagulation factors in a kind of a factor replacement therapy that is at least a traditional therapy. Of course, the protein has a limited half-life in circulation, so you have to keep on re-injecting. And gene therapy has the potential for a much more lasting correction with a single therapeutic intervention. Multiple current clinical trials are all based on essentially the same principle, which is to use an AAV vector with a capsid that has a strong tropism for the liver. This vector is injected intravenously, and it contains an expression cassette with a hepatocyte-specific promoter, ultimately resulting in gene transfer and gene expression in hepatocytes. Hepatocytes produce the missing clotting factors secreted into the circulation, so that now the ability of the blood to clot is restored. And as shown here in this example of clinical trial results, this can result in long-term, even complete correction of hemophilia. However, in some of the clinical trials, transient gene expression was observed, which was correlated to a CD8 T cell response against the capsid and liver injury, as evidenced by the rise of liver enzyme levels. And subsequently, transient immune suppression was incorporated into these trials, using the liver enzymes as a marker for the suspected T cell response. And so once liver enzymes were rising, steroid drugs were administered to suppress the immune response, which was actually successful in several examples in preserving transgene expression. One of the key features was that the response had to be detected early so that the immune suppression could be started right away. We have since attempted to understand how capsid-specific CD8 T cells are activated. And shown here is our current working model, which is based on early findings by Yiping Yang's lab on TLR9-dependent immune responses against AAV, and some of our recent studies, which in aggregate show that the AAV genome is sensed by TLR9, so toll-like receptor 9, which is an endosomal DNA receptor. Sensing occurs in plasmacytoid dendritic cells. These, in turn, produce type 1 interferon, and conventional dendritic cells cooperate with these PDCs to activate CD8 T cells, and that requires sensing of the produced interferon by the interferon receptor on the conventional dendritic cell. And it also requires, additionally, the activity of CD4 T helper cells that provide additional co-stimulatory signals. And so this cooperation between these two different types of dendritic cells, with one being more specialized in innate sensing and cytokine production, and the other one in cross-presentation of antigen, ultimately then activates the MHC1 restricted CD8 T cells. Unmethylated CpG motifs, as they're typical for viral or bacterial DNA, are some of the strongest activators of TLR9 signaling. And so it's been speculated that elimination of such CpG motifs in the AAV genome could reduce TLR9 activation and therefore reduce immune responses in AAV gene transfer. And pioneering work by John Gray has actually led to some of the first CpG-depleted therapeutic AAV genomes. And it turns out that this approach to de-immunizing the AAV vector is actually quite helpful. So it's become clear from clinical trial results looking at the various vectors that were given to patients with hemophilia, that those trials that achieved sustained expression in a larger number of patients utilized vectors that had fewer CpG motifs as opposed to trials that had more patients with merely transient expression. And in these preclinical studies that we've just published, we show in mouse models that indeed using a CpG depleted vector genome results in reduced activation of CD8 T cells against the AAV capsid. However, it does not prevent antibody formation against the capsid, which again is consistent with what's been observed in the clinic. Here's another example of the effect of CpG depletion, this time looking at the CD8 cell response against the transgene product in muscle-directed gene transfer. And what you see is that a conventional vector that contains CpG motifs elicits a CD8 T cell response that infiltrates the muscle and eliminates some of the transduced fibers, whereas a CpG depleted expression cassette can result in more durable expression with less T cell infiltration and less tissue destruction. In this collaborative study between multiple laboratories, we tested a CpG depleted vector genome for expression of human factor IX upon hepatic gene transfer in non-human primates. We utilized a capsid that has strong tropism for human hepatocytes and a factor IX expression cassette that not only was CpG depleted and contained a hepatocyte-specific promoter, but also was codon optimized for expression in hepatocytes, and that contained a factor IX mutation called PADUA that confers a substantially increased factor IX coagulation activity. And as you can see here, is that at quite low vector doses, we were able to achieve high levels of factor IX activity that in one animal shown in the orange, for example, a directed expression of about 25% of normal, and in another animal in 50 to 100% of normal. And so the normal range actually in humans is 50 to 150%. And so if this were a person, we would have corrected the disease to either mild hemophilia or even completely eliminated the coagulation disorder. However, a third animal, as you can see in the depicted by the triangles, failed to express factor IX, and this animal had pre-existing neutralizing antibodies to capsid. So which shows that when you have such an optimal vector that's effective at low vector doses, that you might be more prone to neutralization by even low levels of pre-existing antibodies. However, we are developing capsids that are more resistant to neutralization by pre-existing immunity, and also there are now a number of methods of becoming available to circumvent pre-existing immunity. I want to point out that these three animals had no CD8 T cell response to capsid, no elevation of liver enzyme levels, and did not produce antibodies against human factor IX, so supporting safety of this approach and supporting the use of these CpG depleted vector genomes. At the same time, I would like to point out that not all CD8 T cell responses in AAV gene transfer are TLR9 dependent. There are also TLR9 independent responses, which we found at various combinations of certain vector doses and routes of administration. Besides pathogen-associated molecular patterns, such as the viral genome, there are other activators of innate immunity that are more related to tissue damage, also called damage-associated molecular patterns. And here's a summary of some of those pathways that we have now identified. Some of this work is not published yet, but we believe that in the end, the CD8 T-cell activation occurs through these, again, these cross-presenting conventional dendritic cells, which are XCR1 positive dendritic cells. And their cytokine receptors, for example, sense type 1 interferon that is produced by plasmacytoid dendritic cells that recognize the AAV genome through TLR9. But there are other cytokine receptors, such as the IL-1 receptor, that can sense IL-1 that may be released upon tissue damage. And there are other molecules that can be released upon tissue damage, such as a double-stranded RNA that can be sensed through, for example, toll-like receptor 3. And then ultimately, those signals can activate the XCR1 positive dendritic cell, and in combination with additional co-stimulation through T helper cells, these events will lead to CD8 T-cell activation. Not all activation signals to the immune system are derived from the vector itself, but also come from the tissue. So to mimic this, in this experiment here, we co-administered AAV to skeletal muscle of mice with various agonists to different toll-like receptors, asking whether such AAV extrinsic activation of pattern recognition receptors could impact antigen-specific immune responses, in this case to the transgene product, which was human factor IX. And we interestingly found that co-administration of agonists for TLR2, 4, and 7 had, much to our surprise, no effect at all. We might see inflammation in the muscle, but not necessarily a immune response against the factor IX transgene product. Interestingly, an oligonucleotide that activates TLR9 induced a potent antibody response against human factor IX, which was correlated with an induction of monocyte-derived dendritic cell response. And monocyte-derived dendritic cells, in turn, promote T follicular helper cell responses, which then drive antibody formation. And we further found that this response was modified by regulatory T cells. So for example, if we co-administered the TLR9 agonist and eliminated regulatory T cells, the response deviated away from antibody formation and towards a potent CD8 T cell infiltration of the muscle. So in addition to signals derived from the vector, there can be signals derived from the tissue, and depending on the tissue, it is certain signals, but not others, that can enhance immune responses to the transgene product. And interestingly, regulatory T cells further modulate these responses. In summary, TLR9 signaling in plasma-derived dendritic cells, at least in part driven by unmethylated CpG motifs in the vector genome, results in type 1 interferon-mediated activation of conventional dendritic cells, which cross-prime capsid or transgene product-specific CD8 cells. CpG depletion or use of TLR9 inhibitory sequences, as shown by others, can minimize such responses. Depending on vector dose and route of administration, TLR9 independent CD8 T cell activation may occur. That is because alarm signals from tissue or cell damage, such as release of IL-1, can also induce CD8 T cell responses. Activation of monocyte-derived dendritic cells through vector or tissue-derived signals can promote antibody formation, and regulatory T cells can modulate the adaptive response initially activated by innate signals. I would like to thank the members of our gene and cell therapy program at the Wells Center for Pediatric Research at Indiana University, and I'll be happy to take questions by the audience. Thank you very much.
Thank you, Dr. Herzog. The next presentation will be by Dr. Nagendra Venkata Chemuturi from Takeda and focus on viral gene therapies and pharmacokinetics.
Hello everyone, welcome to my talk on viral gene therapies. At the outset, I would like to thank the organizers, my colleague Halim Cho and Anis, for giving me the opportunity to present in front of you today. In a typical meeting, I would ask through a show of hands those of you who are familiar with viral gene therapies, but that is not possible today, so I'll assume that most of you have a reasonable understanding of what viral gene therapies are about. And in the interest of time, you might see me skip some slides or some bullet points. Please do ask me the questions after my talk is over. We'll be happy to answer them. I have provided additional information, backup slides too. So when you do get a copy of these slides, please do take a look at them. And if you have follow-up questions, feel free to reach me and drop me a line at the email that is provided on this slide. Quick disclosure, I'm an employee of Takeda Pharmaceuticals and I do own common stock in the company. I do not provide any consultancy services to other companies. While the content of these slides have been reviewed within Takeda, the views that are expressed are mine and mine alone and are in no way a reflection of the official position of Takeda Pharmaceuticals Company Limited. So to ensure we are all on the same page as to what gene therapies are, per the FDA, gene therapy products are defined as all products that mediate their effects with the transcription or translation of transferred genetic material, or by specifically altering host genetic sequences. A vector could be viral or non-viral. It could be ex vivo or in vivo gene delivery, integrating or non-integrating, somatic or germline. The objective, of course, is to supply a functional copy of the gene which is responsible for the underlying disease and to provide long, sustained therapeutic effect with single administration. At this particular point in time, due to ethical concerns, germline gene therapies are not being pursued within the industry. Of course, you might read about them in the news as being pursued by individual researchers in certain countries, but definitely not within the pharmaceutical industry. When it comes to the types of gene therapies, as I said, it could be ex vivo or in vivo. I'll be talking about in vivo gene therapies today. It could be somatic or germline. It could be viral or non-viral. When it comes to either the viral or non-viral, they could be both integrating or non-integrating. Non-viral gene therapies are with nanoparticles, lipid nanoparticles, extracellular vesicles, exosomes, etc. The biggest advantage they have, their low immunogenicity potential. What is the biggest disadvantage? They deliver the genetic material to the cytoplasm, and the DNA needs to be delivered to the nucleus, not the cytoplasm, which is the biggest calling card, the unique selling point for viral gene therapies. They can deliver the genetic material to the nucleus. They have evolved over the millennia to do this, to make more and more copies of themselves by integrating into the host genome. At this particular point in time, predominantly, most of the work is focused on adeno-associated viruses or AAVs. They are not pathogenic on their own. They cannot replicate on their own. They do need a helper virus. They do integrate. The wild type AAV does integrate into the host genome, but this feature has been engineered out. The rep and cap genes have been, for the most part, deleted so that they stay episomal and they cannot make more and more copies of themselves. So for my talk today, I'll be focusing it on viral gene delivery, AAV-based viral gene delivery, in vivo gene delivery, focusing on rare diseases, non-oncology indications. So A quick summary on the two products that have been approved so far, Luxturna and Zolgensma. Luxturna is administered directly to the target site of action into the subretinal layers of the eye. Zolgensma is administered through an intravenous infusion, while the target site of action is the CNS. Luxturna is a AAV2 based gene therapy to treat Leber's congenital amaurosis, which is due to the deficiency of a gene which results in the expression of RPE65 protein. The patient suffers from progressive blindness. Of course, once this particular viral gene therapy has been administered to the patients, it kicks in the production of RPE65 protein and they can see better. When it comes to Zolgensma, it is used to treat spinal muscular atrophy due to the deficiency of survival motor neuron 1 protein. Both the products have been approved in multiple countries and are available. If you haven't seen the videos, and I typically show the videos, but today I'm not doing that in the interest of time. If you have not seen those videos, they're very impactful. They do form part of the clinical trial design and the endpoints based on which their approval was based on. So I strongly encourage you to look at them. They tell you about the scope and the impact they have on patient's life. They change the arc of the patient's life. They change the quality of the patient's life, and not only the patient's life, but also the life of their caregivers, their parents, their brothers, their siblings, etc. So please do take a look at them if you haven't already done so. So coming to today's talk, what is there for a pharmacokineticist, and what are my own views on this? Uh, what is the role of DMPK? When I started out in the industry, I was working on small molecules, and through a quirk of fate, I started working with biologics, and then I was working with ADCs, and I thought that itself was pretty intense. And then, of course, when I joined Novartis, my supervisor, Mark Milton, asked me, Nag, I have a lot of work. I'm kind of overloaded. Would you like to take on viral gene therapies? I had to first ask myself, what do I know about viruses and viral gene therapies? And what is the role of DMPK? So there was a lot of reading that I had to do. And initially, I thought there isn't a huge role, but then I learned I was very wrong. There is quite a big role for DMPK and clinical pharmacologists to play when it comes to viral gene therapies. And I'll be taking you through some of these aspects, the most important being biodistribution and immunogenicity. But I'll be focusing on biodistribution because the other talk today is on immunogenicity. And hopefully I'll leave you with a better appreciation for what viral gene therapies are and what is the role of a DMPK clinical pharmacologist professional. When it comes to the role of DMPK and clinical pharmacology, it is different from that of a small molecule or a biotherapeutic, but the underlying objective and the principles are the same. We want to understand the disposition of the molecule. What does the body do to this drug? Of course, these are viruses, so we need to develop newer concepts, maybe newer terminologies, which will more accurately describe this new modality, better disposition, better describe the disposition of this modality. And we might need to develop new models to explain this. What is our role? Capsidum promoter selection, understanding the disposition, which includes biodistribution and shedding, immunogenicity, clinical dose projection, clinical trial design, contribution to the label, the huge bioanalytical package that we need to develop to support these programs. So all these form the role of a DMPK clinical pharmacology professional when it comes to viral gene therapies. So let's look at biodistribution. Biodistribution doesn't exactly look catchy, flashy as immunogenicity or PKPD, modeling and simulation, PPPK, but it is pretty important when it comes to viral gene therapies. When you look at the guidances out there, the FDA has put out a guidance, EMA has put out a guidance. They talk about biodistribution a lot. The IPRP paper, which is a consortium of all the agencies worldwide, together put out a paper on biodistribution of viral gene therapies. That tells you how important it is. And biodistribution is not just the D in ADME, it is the distribution, persistence, and clearance of the vector genome from target and non-target tissues. This is from the FDA guidance, this is from the IPRP guidance, and this is also going to come from the ICH, which is in draft stage at this particular point in time. But why biodistribution? Why not concentration time profiles? And this is something that I had to ask myself when I started out in viral gene therapies. So when you think about it, we are delivering a functional copy of a gene using a viral vector. In the target site of action, this particular gene gets delivered, leads to the expression of a new protein, provides therapeutic benefit to the patient, which is fantastic. On the other hand, when this particular viral vector gets delivered to non-target tissues, can it transduce and express this protein there, where this protein has no function no physiological activity, no physiological function. Can that lead to adverse events? Taken to its extreme, can this be transduced in the gonads, the sperm and the ova, and be passed on to the offspring, which means it is in the zygote and the embryo, which literally means that it could be expressed all over the body. And this is a scene and a scenario which we want to avoid. So this is why we want to look at biodistribution. This is the PK when it comes to viral gene therapies. It is driven by the capsid, the protein code. And it's all about, what it can bind to, which receptor it can bind to. Tropism, the viral tropism could be organism based. That is, all viruses, bacteria do not infect all the species that are out there. They only infect certain species. Within a certain species, they infect only certain tissues or organs. And within a certain tissue or organ, they infect only one or two cell types. I mean, not two, but only certain cell types. Those which carry the cell surface receptors to which these viruses can bind, and that is the tropism. So it is driven by the tropism and not the gene inside. And it is to assess the potential of germ-line transmission, aid in toxicology, understand the biodistribution of these viral gene therapies. It has to be obviously conducted in appropriate genders and appropriate species, transducible species. Kind of akin to, and I'm using a analogy here, the cross-reactivity when we talk about biotherapeutics. You want to make sure whatever your preclinical species is, your viral vector can infect the appropriate cells in the appropriate tissues and lead to the expression of this protein in those particular species. You want to make sure that you are able to capture the peak of the gene therapy production. Of course, these are biodistribution studies, which means the animal usage is high. You are collecting all the tissues that are out there. Maybe you can collect only certain tissues, but have the chat with the FDA. So you want to, the animal usage is high. So you want to make sure you can collect at least 3 time points. Collecting a lot of time points, dense sampling, a lot of time points would mean you have to sacrifice a lot of animals. So you want to keep that to a minimum, keeping in mind 3 hours initiative. Local administration, if feasible, is preferred over systemic administration because it helps ensure higher vector concentration at the site of action and spares the non-target tissues from exposure to the vector, and that's a huge advantage. A lot more information on the conduct of biodistribution studies is provided in slides 22 and 23 in the backup section, so please do take a look at them, and if you have any questions, drop me an email. What are some of the factors which affect biodistribution? Now, please know that, depending on the tropism, biodistribution within a given species is at a macro level, that is distribution to all the major organs through the convective flow of blood. And then it's also at a micro level, that is within a given tissue, how does it distribute? What are the different cell types? It can transduce and lead to the expression of this particular new protein. And that is driven by the capsid, the tropism, and the promoters that are going to be used. What is the route of administration? And that can alter the biodistribution and shedding profile. Such information is provided on slide 26. Once again, local administration, if feasible, is better than systemic administration. What are the different tissues to collect, etc., are provided in the follow-up slides. But when you're thinking about local administration, do think of the draining lymphatics into which these viral vectors could be drained into. You do want to collect them. You do want to collect the gonads, etc. Immunogenicity, etc., could also alter biodistribution profile, and some of the future opportunities like novel imaging techniques, etc., are also discussed on other slides in the backup section. So when it comes to biodistribution, the data does make its way into the label, and I'm comparing and contrasting the biodistribution data that is provided in the label for Luxturna, a locally administered viral gene therapy, and Zolgensma, which is a systemically administered viral gene therapy. Luxturna is administered into the subretinal layers of the eye. So guess what? In NHPs, monkeys, most of the drug is seen in the aqueous and the vitreous humor of the eye, some in the optic chiasm. But outside of the eye, it is mostly seen in the liver and spleen, but that too at very low levels. Vector DNA was not seen in the gonads. Compare that to Zolgensma, which was administered intravenously. Guess what? You do see it in the spleen, heart, pancreas, and lots of other tissues. So much more widespread biodistribution is seen with Zolgensma, which was administered systemically. Moving on from biodistribution to shedding. Shedding is the excretion anatomy. Why is the word shedding used? It has been used historically when talking about the excretion of the viruses, removal of the viruses, intact viruses from the body, and that is what we want to assess. It is about the dissemination of the virus through the secretions or excreta of the patient. Why? is about the contagion, right? An intact virus which is leaving a patient and it can get into another healthy individual and cause disease in those patients. And that is what it is about. We want to understand the potential risk associated with transmission to third parties. Third parties could be the parents, the caregivers, they could be the pharmacist who is constituting the drug, the nurse or the physician who is administering the drug, and the potential risk to the environment. I mean, this is the brand newly engineered viral gene virus, right? It's a new viral vector. And guess what? Once it leaves the patient through his excreta, it could be in the waterways, in the soil, etc., from which it can get into different organisms, the plants, fishes, etc. And we want to know the potential risk to that. Can it get into those species? And in those species, can it infect the tissues in those species? And can it lead to the expression of this protein in those species where it has no role of being present, frankly? That is kind of what we want to understand. The data is used to provide precautions to patients, caregivers, assess environmental risk, etc. And it goes into the label. Do understand that viral gene therapies are considered genetically modified organisms and they have been genetically modified. You're getting rid of their ability to reproduce or replicate, right? So they are reviewed by other departments than just the regulatory agencies in all the countries. Some of the factors to think about when conducting shedding studies. What is the natural route of infection of this particular parental virus? What are the different species it can transduce? What is its pathogenicity, its ability to replicate, pre-existing immunity, the seroprevalence, patient population, etc. You want to think about these when you do look at shedding. It can also be affected by the route of administration and drugs like corticosteroids. What are the different matrices you want to collect when you want to assess shedding? It would be the saliva, nasopharyngeal secretions, tears, feces, urine, etc. And you do look for the vector DNA by PCR. Some of the other aspects of shedding I have provided on slide 32. Please do take a look at it. The data from shedding studies does make its way into the label. Some of the precautions to do with shedding are also provided in the label. And once again, I'm comparing and contrasting the shedding data for Luxturna administered locally into the subretinal layers of the eye, and for Zolgensma, which is administered intravenously. For Luxturna, which is administered directly into the eye, it is mostly shed in the tears, that too transiently at very low levels, and up to 7 to 14 days following administration. We don't really see it in blood, nasal secretions, etc. Other matrices. When it comes to Zolgensma, which is administered by intravenous infusion, it is seen in the saliva, urine, and stools, and that too out to 1 to 2 months. So when it comes to Luxturna, precautions need to be taken for 7 days post administration. When it comes to Zolgensma, you have to take precautions at least for 1 month post administration. What are the precautions that are provided when you are being given Luxturna or Zolgensma? Uh, Patients and caregivers are advised on proper handling of the waste material that is generated from the dressing, tears, nasal secretions, etc. They need to be put in appropriate Ziploc bag type containers, but they can be sealed and they can be disposed of in regular trash. When it comes to Zolgensma, these are babies, so diapers do form a part of this label. They have to be disposed of in appropriate trash bags, could be Ziploc containers too, and they could be discarded in regular trash. Family members, caregivers should observe good hand washing techniques. That is, good hygiene needs to be taken into account. One thing for Luxturna that I don't see with Zolgensma, patients treated with Luxturna should not donate blood, organs, tissues, and cells for transplantation. I did not see similar language for Zolgensma and haven't understood why. Moving on from shedding to immunogenicity, another aspect. These are viruses. So guess what? We will see a host immune response to viruses. You'll see both a humoral immune response and you will also see a cellular immune response. The immune response is not just against the viral capsid, it's also against a newly transduced protein. Take a step back and think about it. This particular patient has never seen a functional copy of this protein. So you're delivering a viral vector which leads to the expression of this new protein, which is foreign to the patient. Yes, it provides therapeutic benefit, but just like with other protein therapeutics that are out there, there is likely to be a both a humoral and cellular immune response against this. So we do want to look for humoral and cellular immunogenicity assays against both the transduced product as well as the viral capsid. But think about this. We are living through a pandemic, right? It's not about where I am. It's about where I have traveled to. Think about the Biogen incident, the Biogen conference incident in Boston. People came in here and then they basically, got infected when they left this place. So it's about where you have been, where you are working, where you are born, etc. Our immune system has a memory. I'll give an example, right? Tetanus virus. They recommend that we take the tetanus vaccine every 10 years. That is how long the immune memory lasts when it comes to tetanus virus. When it comes to hepatitis virus, they ask us to take a booster shot every 5 years. That's how long the immune memory lasts. How long does the immune memory last against a AAV2 vector, AAV9 vector, AAV5 vector? We do not know. I do not know how long the immune memory lasts. But that will definitely help with redosing of these viral vectors, right? Our immune system is primed to recognize these. I travel to some Disneyland wherever with my kids. I get infected with AAV5 viral vector. It's a silent vector. It's not pathogenic. So I do not show any sort of symptoms, but my body's immune system is primed. And if I ever happen to receive AAV5 based gene therapy within the next 6 months, 1 year, however long the immune memory lasts, it might neutralize the viral vector that is being used to provide a gene therapy to me. It might lose efficacy, and this needs to be kept in mind. The impact of immunogenicity is very much route dependent. Now think about Luxturna. It is administered into subretinal layers of the eye, which is a very niche space. Not a lot of blood supply there. So even if the patient's sero levels, against AAV2 vector are really high, it might not impact efficacy as much as it is with Zolgensma, which is administered intravenously. Even a small titer of anti-AAV9 antibodies could reduce efficacy significantly. Not so with Luxturna. So the impact could be very route dependent. Uh, the typical approach is to perform all the possible tests that are out there. But once you have the data, you can always approach the agency saying, this is what the data says. Maybe I don't need to do T cell reactivity against the newly transduced product. And they're pretty receptive to such ideas. It's always a very good collaboration when it comes to viral gene therapies. What does a typical bioanalysis package contain? When you look at biodistribution, you definitely want to measure the vector genome, that is the DNA by qPCR, ddPCR, whatever other technique. But when you want to look at the translatability, which is the mRNA, or the transgene product, which is a protein, maybe yes, maybe no. That depends on the scenario. And please ask me what those scenarios are once my talk is over. I would like to make sure I'm staying on time, by the way. When it comes to immunogenicity, once again, I said humoral and cellular immunogenicity against both the capsid and the newly transduced protein. So that's 4 different assays. When you think about shedding, you just need to measure the vector genome, the DNA. I have also provided the different target tissues you need to measure for biodistribution, for shedding, and for immunogenicity. Do what is appropriate. Measure, those tissues which make strong scientific sense. Do not do and just do not collect all the tissues and do a box checking exercise. Do what is scientifically justifiable, that which has a strong scientific rationale. Other information on bioanalysis is provided, and some bit of PKPD analysis is also provided on slide 30. Take a look when you get some time, and if you have any questions, please do let me know. Drop me a line. Happy to always answer. When it comes to dose selection, how do I determine my first human dose, right? Typical allometric scaling techniques do not exactly apply. I mean, these are viruses, so we need to think of different ways. You want to use all the available preclinical data that is there, including NOL, etc., to come up with the first in human dose. But once you have the dose, once you are able to get into the clinic, dose the first couple of cohorts, look at the data, refine your dose, take it to the agency. The agencies are more than happy to review such data and agree with you or provide their input so that you can refine your dose for the next few cohorts. Maybe there is already a viral gene therapy in the clinic, and you can refine your dose based on the data that is available from such a viral gene therapy, which is still being investigated in the clinic. You can always use modeling and simulation as long as it is appropriate. Uh, Also, if you're going locally, you do want to consider the organ and tissue specific parameters. For example, you're getting into the eye, you want to know the volume of the eye. Maybe you're getting into the vitreous humor, you want to know the volume of the vitreous humor. You want to know how much vitreous humor volume is there in the monkey versus human, etc. Use appropriate scaling factors when you're going locally. What are some of the current challenges with viral gene therapies? Durability. We don't know still how durable and how long therapeutic effect lasts. The LTFU does provide you with some guidance along these lines, and you do want to follow the patients anywhere from 5 to 15 years to ensure efficacy and make sure they are not suffering any hidden long-term adverse events. The dose, once again, once you have dosed these viral gene therapies, you cannot abrogate or nullify them. I mean, maybe you can give antiviral capsid antibodies, etc. But once they have been transduced, it's kind of hard to undo it. Sometimes the dosing is pretty invasive, so you want to keep that in mind. Redosing is a concern due to immunogenicity, but folks are investigating that. Hot topic for investigation, as the previous talk hinted. Manufacturability, CMC is the biggest hurdle still. And these are rare diseases that I'm talking about, right? The patients are rare, they're spread out geographically. And a lot of times the patient advocacy groups, foundations, disease foundations help you fly them in to the clinical trial site, house them there anywhere from 6 months to 1 year at times. So you do want to work with partners like the Bespoke Consortium, the disease advocacy groups, and disease foundations to help with the conduct of the clinical trial and monitoring the patients. What does the future hold for viral gene therapies and for a pharmacokineticist? Integrating gene therapies, other viral vectors like the adenoviruses and lentiviruses, dual vectors, non-viral vectors are also pretty hot right now. Immunosuppression and immunomodulation techniques to enable redosing of viral gene therapies is being looked at. And once again, we need to understand the immune memory that lasts. As I mentioned, tetanus 10 years, hepatitis C 5 years, COVID-19, they're asking you to take a booster shot like 6 months or 9 months after your second dose. So these are some of the things we need to think about when we think about redosing of viral gene therapies. Combination viral gene therapies, taking a big gene and breaking it into 2 or 3 pieces and redosing them. Please do understand that it's a pretty hot area. It's moving very fast, evolving very fast. How about for a pharmacokineticist, DMPK, clinical pharmacologist? Novel bioanalytical techniques, especially imaging techniques, which can help reduce the use of animals. In silico modeling, which can predict biodistribution and decrease immunogenicity. Once again, anything that can help reduce the use of animals would be fantastic. QSP models, which can, once again, predict biodistribution, predict the durability of the therapeutic effect, immunogenicity should be used. In silico techniques to identify, immunogenic epitopes on the protein can be used to basically, modify the gene to decrease the immunogenicity of the protein itself. Engineered proteins, you have this protein which is present in healthy individuals. Why not come up with a protein which is even better than that? That will provide a much better therapeutic effect to the patients, kind of like Hemlibra. If you have already heard of Hemlibra, you know what it is. If not, ask me after the talk. And then drug-drug interactions would be a huge topic going into the future. So when it comes to gene therapies, no one size fits all. That's my one piece of advice to you. It's a rapidly evolving field, lots of developments, and it's a bit hard to keep on top of everything that's going on. New areas, new vistas are being explored and investigated. And with that, the role of DMPK and a clinical pharmacologist is evolving. It's expanding. A lot of opportunities, but there could be a lot of pitfalls too. So one needs to watch out. And no one size fits all. Doing what is appropriate with a strong scientific rationale is the right way to chart the course. Tailor studies based on the route of administration, the viral vector, the risk assessment, target indication, etc., the patient population. Do not conduct a study just because you can do it. Conduct a study with a strong scientific rationale, strong justification. If you go to the agency and say you will not collect all the umpty number of tissues that are mentioned in the guidance, they'll be happy to listen to you. But if you provide a strong scientific rationale, you'll be surprised. They'll be happy to agree with you, in fact. Once again, these are rare diseases, a very underserved population. So the agencies are more than happy to provide you with really good input, act as partners and collaborators. They are learning, we are learning at the same time. So do what is relevant with strong scientific rationale. Avoid box checking exercises. Seek agency input, and you can seek it pretty often too. So that's my message for you today. And with that, I would like to end my talk and acknowledge my supervisors, Nisha, Mark Chen, Vinci Shu, Hailim Show, and Novartis Institutes of Biomedical Research and BioProcess International. And with that, I'm happy to take any questions that you have today.
Okay, great. Thanks, Dr. Herzog and Dr. Chemuturi. So the first question is, how much does the AAV serotype versus the target tissue matter for the risk of immune responses?
So maybe I can address this question. Yes. So, I mean, in general, the vector dose is going to be important for how much of an immune response you get to, for example, the viral capsid, and how much DNA input there is that could be sensed by the innate immune system, right? And that's going to matter in the context of the tissue that it's being injected. But at the same time, the serotype will determine not only tissue tropism, but also how much gene transfer there is to the target cells. And so a certain combination of dose and capsid or serotype will give you a certain amount of transgene expression, which is, of course, then further modulated by your choice of promoter and so forth. And so, so it's going to have an impact on the level of transgene expression. And the level of transgene expression, as we learned, does have a substantial impact on the likelihood of immune responses to the transgene product, to your therapeutic product that's being expressed. So how much of that you express in the context of a particular tissue will determine whether there is an immune response or not, or in some instances even whether you might even able to achieve immune tolerance to your therapeutic product.
Okay. And while immunomodulatory techniques are being attempted, would it ever be possible due to immunogenicity concerns? And can you provide your thoughts on this topic? I think maybe for Nag.
I would let Dr. Herzog go first, and then I could go next. I mean, since he is the one who gave the talk on immunogenicity, I would be interested in listening to his thoughts. Dr. Herzog?
Yes, I believe the question was about immune modulatory protocols.
Yes.
Yep.
Yeah, I mean, they're already included in many designs, clinical trial designs. And I suppose looking across the various clinical trials, what's most commonly used are steroid drugs. In some instances, they're used because of the underlying disease that is being treated with gene therapy is prone to inflammation and tissue regeneration, and so to dampen the underlying of inflammation at the time of gene transfer, steroids are used to counter that. In other cases, steroids are used as on standby, so that if there is an immune response or a biomarker for an immune response is measured, and then steroids are administered to counter, for example, T-cell responses to the viral capsid. And I think beyond that, the field is working toward more targeted interventions, and so it's already part of clinical trial design these days, there are new protocols emerging, such as combinations of transiently depleting B cells combined with a T-cell targeting agent and so forth. So the field, I think, is learning how to combine at least transient immune suppression with gene transfer in a smart way in order to eliminate some of the potential immune responses or immunotoxicities.
I agree with Dr. Herzog. Whatever he has said. Immunogenicity seems to, if I can borrow from Shrek, immunogenicity has layers, like onions have layers. It doesn't seem straightforward. When you think about some of the data that is coming out of the clinic from Unicure and BioMarin, I do find it interesting, and I would love to hear Dr. Herzog's thoughts on this too. But when it comes to AAV5, it doesn't seem to affect the neutralizing antibodies against the AAV5 capsid, do not seem to affect the efficacy as much as a neutralizing antibody against AAV8, AAV9 would. And this is something we are learning. Another aspect that has been published on is that some of the binding antibodies against the capsid could actually increase efficacy. And I have seen something like this, a very similar effect like this with antibody drug conjugates in my previous career in oncology. But this is another aspect which, as I said, there are different layers to it. It doesn't always seem straightforward. And what do we do when we think about immunomodulation, immunosuppression, etc.? Looking at the data helps, and some of what Dr. Herzog says definitely helps.
Okay. And can you design an AAV vector so that no immune suppression is needed?
I think it depends on so many factors, like I mentioned before, dose, route of administration, and so you can, I think, engineer the virus to really minimize innate immunity and also the potential for the virus to accidentally express in the transgene in antigen-presenting cells, and so there's a number of things you can do to reduce immunogenicity. But depending on your dose and your route of administration, it may not be possible to make a virus that is just so stealthy to the immune system that you don't need to do anything else. Again, depends on your applications, but I think that for many applications, in the end, there may be some where the virus can be engineered and it's sufficient for your purpose, and you don't need to further immune suppress. But I think there'll be many other applications where you will have to where those two approaches could be synergistic. So you take away some of the immunogenicity of the virus through genome and capsid engineering, but you still have to have a supplementary kind of immune modulatory regimen, because you may just not be able to entirely eliminate all the responses that you're concerned about.
Yep, I mean, you can never ever, these are viruses, so completely abrogating immunogenicity or host immune responses, I don't think that's possible. I mean, attenuate, decrease, yes. I mean, we have immunogenicity against PEGs, polyethylene glycol polymers. So getting rid of the immunogenicity completely against a virus, I think, is out of the question.
Yeah. I think, when in the ocular gene transfer, if it's a low amount of virus that you engineered properly and it's a low dose and you're going into an immune privileged site and the expression persists, you might just be fine as in the LCA treatment. But for many other applications, that just doesn't apply. You may just not have an immune privileged site, or you might have to give much more of the virus and so forth. And then I agree. Then immune system knows how to fend off viruses, so you're still going to have to do something else alongside your gene transfer.
Okay. I think we have time for just one more question. If we didn't get to your question, we'll be sending those directly to the presenters, and they can follow up with you via email. So go ahead and continue to type in your questions if you still have them. The last question for the presentation, though, is there anything being done regarding trying these therapies in sickle cell disease that you know of?
Sorry, sickle cell disease and what else? I didn't hear that.
That was it. Do you know of any research being done for gene therapies for sickle cell disease?
I think there are trials. Sorry, go ahead, Dr. Herzog.
Yeah, there are multiple clinical trials, and some of them are based on lentiviral gene transfer to hematopoietic stem cells. Some of them now start using gene editing technologies. There's actually a lot of hope that gene therapy for sickle cell disease will be successful.
I agree. There are multiple clinical trials, multiple companies looking at it. I think just last month, Novartis had announced a collaboration with the Bill and Melinda Gates Foundation to look into sickle cell disease again. So, yep, multiple shots at the goal.
Fantastic. Okay, well, thank you, Dr. Herzog and Dr. Chemuturi.
Yeah, thank you.
And
thank you.
And thank you to our audience for joining us. The recorded version of this webcast will be made available for on-demand viewing on our website. And as a registered attendee, you'll receive a follow-up email providing us a direct link. We look forward to having you join us at future BioProcess International Ask the Expert webcasts. Look for those announcements in your inbox. Goodbye.
Thank you. Bye.
Bye-bye.